Welcome, I'm Ann Vandersteel and I'm joined with Matt Godovin from Vitanova Biomedical. Vitanova is an early-stage biotech firm and they are based in San Antonio, Texas, where they've made some startling discoveries in the exciting world of cancer research. It's a pleasure to have you here today. Thank you so much for joining us. And let's take a quick deep dive about what you have discovered and tell us how this is going to impact the patients out there that are suffering terribly from cancer.
In my research lab at UT San Antonio, I've developed a technique that takes a compound and a wavelength of light. So we're making targeted compounds that will seek out a patient's cancer tumor. Once that compound is brought to the tumor by injection into the bloodstream, it's internalized. We then externally activate that particle with a beam of light. That activation will cause an acidosis that's just inside the cancer cells that's so rapid and so extreme that the cancer cells rapidly kill themselves. And we've demonstrated this to be true for 5 different types of cancers.
Okay, which ones?
2 forms of breast cancer, including triple-negative breast cancer, 2 forms of prostate cancer, and with pancreatic cancer.
Wow, that is extraordinary. Tell me, where are you doing the research right now? It sounds like you're still in San Antonio. Is it based out of the university?
That's correct, yeah. Currently
Okay.
I'm doing the research in my lab at the university, and I've created the company, Vitanova Biomedical, to commercialize the technology and bring this to phase 1 clinical trials for patients.
Very exciting. How long has Vitanova Biomedical been in business?
I formed the company in 2014 by myself with my chief financial officer, and then brought on Tom Roberts, our CEO, a little over a year ago.
Fantastic. And what is Tom bringing to the equation for your company? How are you getting the company to move forward? It seems like you have got a lot on the plate. You're preclinical right now, which means you have kind of a long way to go, which I presume means you have to raise money.
That's correct, yeah. And so I think we have sort of this perfect marriage between the science expertise that I bring from the laboratory and Tom over 15 years of expertise in business with different companies and former CEOs of startups, and especially those that come out of academia. So he's just got a wealth of expertise, and he's really driven us to where we are now.
So tell me a little bit about the research area. Currently you are injecting cancer into guinea pigs, and you are proving that you can actually kill that cancer. Is that correct?
That's correct, yeah. We have different animal models, but we will inject, for example, we injected triple-negative breast cancer into the chest of a mouse, and then with one treatment showed we could significantly shrink the tumor and extend the animal's life significantly.
Wow, that's incredible. Absolutely incredible. Cancer impacts so many people. I'm actually a 2-time survivor myself. My parents passed. I have a lot of friends with cancer. This is not something that people can get their hands on today.
No, it's correct. One of the things that we are excited to sort of share with everybody is this concept that this is a platform technology. So because we can kill any kind of cancer, that we can get this particle, and that's really only our only limitation. We have ideas to treat prostate, breast, lung. We have a whole laundry list. All we have to do is reformulate the particle. But the basic core technology is the same, and it's that basic core technology of light-activated acidosis that the company has the intellectual property on.
Very impressive. So tell me, by utilizing technology like this, does this mean that people who have inoperable cancer can actually be saved potentially in the future, or people that don't want to endure surgery because of the negative effects, physical harm, disfiguration, etc.?
Yeah, I mean, so some patients aren't even offered surgery if they have comorbidity factors, if their blood pressure isn't in control. They just can't have surgery.
Right.
Sometimes they can have surgery, but the tumor is in a place that's inoperable. And so we feel like since the compound by itself does nothing harmful and the light by itself does nothing harmful, it's only when these two things are really focused and focally put together do they cause this rapid cell death. So yeah, we feel like this could be offered to people that can't tolerate radiation, maybe aren't having luck with the chemotherapy and success. Sometimes chemotherapy, patients become resistant to that, and also for patients that either can't have surgery or have a tumor that's in an inoperable location.
I understand you're looking to raise about $1.5 million. What is the time period that money will be used, and what is your staging for financing into the future as you go from a new drug application to becoming a new drug compound?
So right now what we're doing in our first seed round is looking for funds to deliver 2 measurable objectives to investors, and that is show that the nanoparticle will be formulated by one of our strategic partners that will analyze all of the stability, shelf life aspects of it. That'll be the particle that'll be used in all the subsequent studies. So product formulation, and then we have small animal studies that will be prove out the efficacy for the FDA. So those two would be the two milestones that we would deliver with the $1.5 million. It's going to take about $3.5 million to get us to conduct the pharmacokinetics, pharmacodynamics, toxicology, and large animal studies that are required for the investigational new drug application. So we have some federal grants that we've applied for, so we don't know exactly how much of that $3.5 million we would need. We know the $1.5 million would get us to that point. It would be about a 12-month endeavor. We'd start looking either for those grants to kick in or go back to another seed round.
Okay, so for this $1.5 million round right now, it's going to be a convertible debt note that could potentially become equity in the company at some point in the future. Is that correct?
Right, yeah, we anticipate that we would, even in multiple seed rounds, that we would offer the investors convertible notes. All the notes would be converted at the point of valuation at Series A funding.
Is anybody else doing this sort of targeted therapy that you're doing? Has anybody else come close to this new technology that you have developed?
Some of the therapies, some of the immunotherapies are targeted. They have the potential to have devastating side effects, though. So one of the things we're really excited about is the fact that in our animal studies, the animals showed no signs of any pathophysiology in response to the treatment. They were grooming, eating, gaining weight. And so we want to be able to offer that to cancer patients and for those that care for them.
So essentially this is a targeted therapy. It's an immunotherapy. It's not surgical. You're using two separate entities, light and your compound, married together, and independently they're not harmful. You put them together and you put the light on it. It actually kills the cancer cell, creates acidosis, and the cell dies from the inside out and does nothing else to be harmful to any of the surrounding cells, like chemotherapy, like radiation.
Correct.
Wonderful.
Right, and the idea that this is a platform technology says that all we have to do is take the compound, the nanoparticle, and put a different when we sort of talk about the cancer has a receptor,
right,
and our particle seeks that out and binds to it. So prostate cancer has a perfect antibody-antigen pairing. So what we do for breast cancer is find a different receptor, take the same core technology, and inject that. That should seek out receptors on breast cancer, ovarian cancer, brain cancer. We want to be able to make this a platform technology.
So exciting, Matt. It's just absolutely extraordinary. And like I said, being a survivor and knowing so many people, cancer is really not selective, is it? It's just that disease that's everywhere, and every day you hear of somebody else that's being impacted or affected, family members, etc. So it's very exciting to talk to you today.
Thanks.
I'm really excited about this. I would like everybody to, take a look at your due diligence report that you should have in your hands, which has Matt Godovin's name and contact information, as well as the company information and the CEO's information, where you can learn more about the health of this company and the exciting information that they are forthcoming. So thanks so much for joining us today. We appreciate it. Matt, it's been a pleasure having you here, and I'm very excited for what you're bringing to the table for cancer. This is going to be history in the making.
Thank you so much.
Light-activated intracellular acidosis is a new form of cancer therapy involving near-infrared light. In order to deliver light-activated intracellular acidosis to a patient's tumor, an upconverting nanoparticle is created, which adheres the photosensitizing compound to the surface of the nanoparticle. A layer of protein is added in order to attach a specialized compound to the surface of the nanoparticle. This compound will seek out and attach to receptors found on the cancer cell surface. This targeted nanoparticle is injected into the patient, and it will travel through the bloodstream and eventually reach the site of the tumor. The nanoparticle will then leak out of permeable vessels near the tumor. It will bind to special receptors on the surface of cancer cells. Once the conjugate is bound to the cancer cell, the nanoparticle is brought into the cancer cell. The nanoparticle is then activated by deep penetrating near-infrared light. When the nanoparticle has been activated, it causes an immediate intracellular acidosis just to the inside of the targeted cancer cells that receive the near-infrared light. The cancer cells cannot overcome the light-induced intracellular acidosis, and a programmed cell suicide or apoptosis begins. Nearby healthy cells without the special receptors will not receive the drug or the light and will not be affected. Apoptosis triggers the immune system to remove the dead cancer cells. The tumor will die and normal tissue will regrow in its place. Light-activated intracellular acidosis is a new form of cancer therapy that combines 3 proven technologies: the effectiveness of intracellular acidosis to kill cancer, the ability to use specific wavelengths of light to activate compounds, and active targeting of nanoparticles specific to cancer, thereby sparing healthy cells.